Hello, everyone. Welcome to another episode of Research Digest podcast. In this episode, I have Dr. Juliet Obi. Juliet is currently working at Eli Lilly, a very famous and global biotech company. And I have known Juliet for a long time, and I know she has been doing a really good work. Uh, and she has done some work on dengue virus and some molecular biology and structural biology stuff on dengue virus. So we'll talk about all those stuff. First, Juliet, welcome to this podcast.
Thank you so much. Thank you for having me.
Yeah. So I know about you, but for our audience, please can you share your journey so far in the academia and industry?
Yes, so like Sam said, my name is Juliet, Dr. Juliet Obi. I recently, 6 months ago now, I recently graduated with my PhD in pharmaceutical sciences in May. And I did that from the University of Maryland, Baltimore. And yeah, so before that, I also got a master's in pharmaceutical sciences, and I got that at Albany College of Pharmacy, which at the time it had two campuses. So I actually got it at the Vermont campus. So I was in Vermont for 2 years before I moved to Baltimore for my PhD. So yeah, I'll say that's just a quick summary about me. I'm sure we're gonna get into it tomorrow as we go.
Right. And what are you doing now, like briefly?
Yeah, oh yeah. So I'm currently a PhD level scientist. My title is actually advisor, which is the, I guess, title for the first, I guess, entry level for PhDs
Oh,
in at Eli Lilly and Company,
mm,
whose headquarters is in Indianapolis, in Indiana. But I am specifically in the Biotechnology Center, which is here in San Diego, California. So I am fully on site for my role. So yeah, so that's my role. Again, we're going to talk more about my role, but yeah, that's what I do. I'm an advisor in biophysical chemistry here in Lilly, and I started that in June. So it's been 6 months now.
yeah, that's great. All right. So let's talk about little bit on the science part of your journey. So I know you are working on dengue virus,
Mhm.
especially a non-structural protein of dengue virus. So we'll talk about your wonderful article that you have published this year in Nature Publishing Journal Viruses.
Mhm.
And before going into particular research findings, can you share with us about current danger of dengue virus, like kind of the epidemiological aspect?
Yeah,
Yeah.
yeah. Yeah, so dengue virus is the virus that causes dengue fever.
Mhm.
And I guess for those that will be listening to this, dengue fever is basically the most prevalent mosquito-borne virus. And it mostly affects people in tropical and subtropical regions. So we're talking places like Sub-Saharan Africa, Southeast Asia, and also the south of the Americas. And so that's where it's mostly prevalent.
Yeah.
And
Another, if I can add, another most prevalent mosquito-borne disease is malaria, right? That's
yes,
another.
malaria is most prevalent in I would say it's in sub-Saharan Africa,
Okay. Mhm.
Southeast Asia, but like globally,
Globally,
dengue,
dengue is more prevalent. Mm.
yes, dengue is actually, which is it's a very interesting fact because when I started working on this, um, I was like, no, there's no way malaria is it's malaria, Um, but globally, dengue is. I think the problem is that dengue is just it's more understudied.
Mhm.
And even though there's not a lot of data on dengue cases in places like sub-Saharan Africa, it's there. It's just not well documented, and also because of the symptoms. The symptoms are very similar to malaria. So, for a country like Nigeria, where I am from, you have symptoms, you probably think it's malaria first. You wouldn't think dengue
Hmm. Okay.
Yeah. So the danger of this fever of this virus is the virus is very interesting. I think that's also one of the reasons why I was very excited on working on this project for my PhD. So the virus itself, there are four types or serotypes,
Hmm.
basically, and that depends on kind of like the antigens that are presented when the virus affects the human body. And not only is the fact that there are four types, the issue is that those four viruses, it's the same dengue virus, but they behave like four distinct viruses. And
Okay.
so basically you can be infected up to four times.
Hmm.
And so you can already imagine when you're trying to attack this from the therapeutic or vaccine point of view. So from
Hmm.
the vaccine point of view, the issue with that is that you're going to have to find a vaccine that can hit all four. But
Okay.
right now, there is no vaccine. There's no effective vaccine that can hit all four.
Okay.
And the other issue is that there's a phenomenon called antibody dependence enhancement. And that is basically a phenomenon where when one is infected with one type of the virus, and then you happen to be infected with the second type of the virus, what happens is that because the second type, your immune system does not recognize them, doesn't remember them, and what happens
Hmm.
is the virus actually attacks the immune system itself. It attacks macrophages, and then it actually
Oh,
uses macrophages to actually to internalize itself into the cell, and that basically increases the viral load, and it leads to severe dengue. So in children, it's more fatal, and so that's the issue with this virus. So it's a problem from the vaccine point of view, also from the therapeutic point of
right.
view. There are vaccines, but there are vaccines for specific serotypes. So you do have to get a test for your serotype to be checked before you can even get the vaccine. The vaccine is also limited to people between
Yeah.
9 and 16 years. So you
So
see
the
all of this.
okay, yeah, interesting. So the vaccine that's available against which particular serotype?
I think it's serotype 2.
Okay.
Or maybe it's 3, I forget now. But it's a specific serotype.
Okay. And out of these four serotypes, is there like one of them is more prevalent than others? Is okay.
Yeah, so 2 and 3
Okay.
are the
Hmm.
most, and I think serotype 2 is also the most pathogenic.
Okay, I understand. Okay, so you talked about the vaccine perspective. There is one, but that one is not effective against the other. You mentioned about the vaccine part. So how about the antiviral therapeutics? Is there any effective therapeutics against any of those serotypes?
Yes, good question. So yeah, unfortunately, there is no antiviral therapies in the clinic yet. So none of them have passed the clinical stage. There's some that have gotten to preclinical, but none of them have actually even entered the clinical stage. So there's nothing as at now. So from the therapeutic point of view, there's still a lot of work to be done. And the reason why this virus or this disease is of concern, according to the WHO, is actually in the list of emerging pathogens. And the
Hmm.
reason for that is that because obviously these are mosquitoes, right? So mosquitoes migrate. They like warmer temperatures. Um, they like specific places where they breed. So if you enable those environments, they will grow. And obviously, we know that uh, what's it called? Global warming is an issue. And so, because of all these issues with climate change, even in the US, there are actually cases now that have been detected in Florida. This is like Florida and Texas. There are actually some cases of dengue there. So, there is a projection that, in the next 30 to 50 years, we're going to start seeing more and more cases of dengue, even in the US. So that's why it's really something to be tackled now more than ever so that we don't have an issue like COVID.
Got it. Yeah,
Yeah.
that's really important to plan ahead.
Yeah.
And so you are working on a particular viral protein NS5.
Yeah.
Yeah, tell us more about that. Why that NS5 is important, and
Yeah.
what have you done so far?
Okay. So yeah, so for my PhD, I was tackling this virus from the therapeutic point of view. So like you said, the protein that I was studying is non-structural 5 protein. So
Hmm.
not to get into the nitty-gritty details, but the dengue virus is an RNA virus. And so it's a positive sense RNA virus as well. So once it infects the cell, it's ready to basically be translated into the different viral proteins. So one of the viral proteins is NS5.
Hmm.
And the reason why NS5 is important is that one is the largest out of the non-structural proteins that the virus makes or it has. Also, it has the polymers. So the NS5 is actually a combination of the methyltransferase, which is important for certain things like capping, methylation, which is important for the integrity of the viral genome. And then
Hmm.
that methyltransferase domain is fused to the RNA-dependent RNA polymerase, the RDLP region. So the
Hmm.
polymerase is responsible for the replication of the virus. So for most viruses, or if not all viruses, their polymerases are literally very important. So
Yeah, kind of
basically,
essential for their life cycle.
exactly. So that's why the NS5 protein is a very attractive target. And also because it's also basically the NS5 is a combination of the polymerase and the methyltransferase. Basically, you can look at it like, okay, I want to try and target the polymerase or maybe target both. So my
Hmm.
PhD was actually looking at how we can target, see how we can look at it from the two domain perspective and see how we can target both. Because from my PhD, the work I did, I found out, and I guess that kind of leads me into the work that was published in NPG viruses. Is that we saw that these two domains they actually talk to each other. They are quite flexible and they communicate with each other. They kind of do work together. So the polymerase, of course, does replicate the virus, but there's a lot of dynamics happening between the two. And I think by evolution, that's probably why they are fused together, because a lot of viruses actually have their polymerase separately and their methyltransferase separately. But it's
Hmm.
interesting that the dengue virus has both of them fused together. So probably by evolution, the virus has evolved to kind of make it work where it's more efficient by
Hmm.
these two, working together. So yeah, so that's what I was doing, and to get more into it, I was basically looking at the interactions of the NS5 protein with different RNA substrates because during the replication cycle, it has to NS5 has to interact with different RNA substrates to replicate the virus. So one of the ones I really worked on
Hmm.
is one called stem loop A, and so stem loop A is basically it's the one that initiates viral replication. It's the
Yeah.
promoter for viral replication.
Yeah. So a very important part of your work is um, investigating this particular interaction between this RNA, stem loop A, and the NS5. So, yeah, tell us more about that interaction. Why this RNA is important. Yeah.
Yes. So the RNA is important. So again, NS5. So if we look, we have to be able to understand how important it is. We have to actually go or kind of address the life cycle of the virus.
Hmm.
So like these viruses, they are very smart. They have a way of doing their things. So normally, when the virus infects the cell, it first of all makes it makes the first couple of proteins, but then it has to go in and make more. So NS5 has to be made, and then when NS5 is made, then NS5 has to go in and use the cell and host cells machinery to make more viruses. But before it can start that, it has to first interact with this particular RNA substrate called stem loop A. Now, the interesting thing is that viral replication, so the viral genome, it goes from a 5 to 3 direction. So the viral replication starts on the 3 prime end of the viral genome. But this particular stem loop A is actually located in the 5 prime end. So there's a major conformational change that happens. So and so there are people that actually study that. There are groups that study so it's called the cyclization of the genome. There's a whole study on that. And the genome cyclization involves a lot of RNA interactions. So there are lots of groups that focus on that. I focused on the specific interactions between this protein and that particular stem loop A. So stem loop A basically it kind of serves as a recognition site. And so
Mm.
once the NS5 interacts, recognizes it, it can then cyclize and then go back to the basically the three prime end, and then you can start the bioapplication. So when I started my PhD,
Mm.
what was known in the field at the time was that the polymerase domain in NS5 was the one responsible for interacting with stem loop A.
Okay.
So when I started my PhD, I started with my preliminary studies, and my preliminary studies were kind of saying otherwise. They were pointing to the fact that no, both of the domains, both the polymerase and the methyltransferase domains, are involved in this interaction, and that's because when I did surface plasmon resonance binding studies, and I did the full length NS5 protein, the binding was pretty tight. And then I actually expressed and purified the individual domains, and I still saw
Mm.
interactions. Yes, the interactions were different to varying degrees, but there were still interactions. And then I also used mass spectrometry, specifically hydrogen deuterium exchange, which studies
Okay.
protein dynamics, and
Mm.
it helps you to kind of understand on the protein if there is any interaction. So I did that, and that was where I got my answer. I actually saw that both domains are involved in this interaction, and there's even a
Mm.
lot of informational dynamics that are involved in these interactions. So that was, I would say, that was a very big observation for my PhD, and that kind of like moved, at least for me, I'm happy that I kind of helped advance the field, what we know now.
Yeah.
And so when you are trying to look at it from a structure-based drug design perspective, you should take that into consideration. So you can actually target maybe regions of these interactions between the two domains
Mm.
by a drug to be able to hit that. Because the other thing I did was to then do structural elucidation of the complex. So at the
Yeah.
time as well, we didn't have proof that there was an actual interaction, and the way to obviously get proof was a structure, right? So that kind of got me into the world of cryo EM, and then with cryo EM, we're able to um, get a structure of the complex. But what was interesting is that actually before I got my structure, another group in Japan has actually they actually released their own study. But
Okay.
thankfully, yeah, so they were the first. So I unfortunately I cannot brag. I was hoping to have bragging rights that I
Uh,
was the
okay. But anyway, you have done your part. You didn't know that someone else is working.
Well,
So,
and yes, the good thing is that they were working on a different serotype anyway. They were working on serotype 3, and so they were actually the first to come up with the structure. And that made me happy, at least that made me happy that, okay, everything I was doing right is correct. So I still
mm.
went ahead with my own, and so with serotype 2, I did that, and then I was able to get a structure. And so now we know, and then a lot of what I did also in that paper is also kind of like giving me more information for structure-based design, which was the second part of my
Mm.
project, to see how we can rationally target the protein with small molecules. And the way to target it is to take advantage of what we know about these interactions with not just stem loop A, but even other RNA substrates, and then target those. You can even take advantage of the dynamics. Sometimes you can target some sort of interaction. Maybe there's a movement in the protein, right? The conformation
Mm. Yeah.
if you can lock that movement with a small molecule, then you kind of you render that interaction incompetent or RNA incompetent interaction. So those are the kind of things that I did, and that was the basis of the paper that came out earlier this year.
Yeah. A, a lot of observations, important findings. I just, yeah, that's the beauty of structure that we can kind of develop structure based therapeutics based on those interactions. And another thing I want to point out that in your cryo structure with
Mhm.
the protein and RNA, you mentioned that this interaction is conserved among different serotypes. So I guess in for the future, there it might be possible to develop an antiviral drug that will work against multiple serotypes, right?
Yes, exactly. That's exactly the, or I think that's the goal. Is like if we can actually find, because one of the things I was doing for my PhD, I worked on serotype 2. I mean, obviously, it's a PhD. I'm not going to be there for 12 years. I did my part, but the
But
idea
I'm
is
sure
to,
other people are working on other viruses.
yes. And even in terms of just structure, what we know about the structure of just the protein by itself, we actually, as at today, there are structures of serotype 2 and serotype 3 NS5, the protein by itself. There are no structures of serotype 1 and serotype 4 right
Mm,
now. So we still have, or there's still some work to be done, because another thing to understand is, and I guess I didn't mention that, is that within the serotypes, the structure of NS5 within the serotypes is actually conformationally diverse. So we have open and closed conformations. So serotype 2 assumes an open conformation, serotype 3 assumes a closed conformation. But what I actually found out as well is that even a serotype and serotype 2 can actually sample both open and closed conformation.
okay.
So we actually have structures in open conformation. For example, this virus also, it not only targets or interacts with RNA substrate, it also interacts with the host cell's immune factors. So it
Mm,
interacts with a protein like STAT2. STAT2 is very important in the immune system, signaling and making sure that, um, you know,
okay.
yeah, so this protein directly interacts with STAT2 to actually target it for degradation.
Okay.
And there is the cryo-EM structure of
Mm,
the protein STAT2, the same serotype 2, but it is in the open conformation. When I
mm,
saw the cryo-EM structure of this same serotype with the RNA, it was in the closed conformation. So I think it also uses the conformational diversity to kind of confuse us and
mm.
make it very difficult to come up with a drug. But I think we need to find a common mechanism. That's what we need to find. Some sort of common mechanism that it uses.
At this point, is it known like which conformation does what during infection?
No, right now it's not known. It's still something that I guess we're still trying to pass out.
Mm,
It does seem like it prefers the open conformation for interacting with STAT2.
okay.
There are other, I think that's the thing. There needs to be more structural elucidation of this protein with other immune factors. And then there needs to be even more structural elucidation of the protein with the RNA substrate. Because stem loop A is just a promoter of viral replication. You
Mm,
need the structures with the actual viral genome, which we don't have yet.
okay. I understand.
So there's a lot to still be done.
I understand. Yeah. Hopefully, other scientists will carry on the research and will
I
get
hope so.
ready. Yeah. Yeah.
I hope so.
So, thank you for sharing your research. Let's move on to your career part. So, currently, in your role at Eli Lilly, what do you do in your day-to-day life? And what's yeah.
Yeah, so in my role, my role is specifically I support AI machine learning efforts. And
Mm,
specifically what I do is basically we're trying to obviously we all know where the field is, this is across all pharmaceutical biotechnology
mm,
is everybody's trying to ramp up their AI and machine learning efforts, right?
yeah, yeah.
That's that's the future. We know that. So I guess in Eli Lilly's bid to do that, they're really, and it's been in the news. We all seen it. That's something that they're really trying to ramp up. And so specifically in the Biotech Center here in San Diego, they have an AI and machine learning or computational group, and so basically, there are a lot of efforts which are integrated efforts between the computational group and the experimental group. And so the idea is to ramp up their efforts. And so to be
Mm,
able to ramp up their efforts, they also have to ramp up the assays that they use to characterize all of these biologics. So when you look at it from the biophysical point of view, they
mm,
have assays that they use to characterize these antibodies. So specifically, it's antibodies or it's biologics that they
Mm.
focus on here in the Biotech Center in San Diego. So there are developability assays that they use to characterize them. You may characterize the hydrophobicity. You can want to characterize the surface charge. You want to characterize the um, what's it called, the aggregation states, aggregation level. So you have to know all of that because those are very important for formulation down the line, right? So basically, a lot of those developability assays, they need to ramp it up. Specifically, they need to kind of implement higher throughput assays. And so
Okay.
that's where I come in. So my role is to help to one, bring in higher throughput assays, not necessarily to replace the ones they have, but to support or to augment it specifically for these AI machine learning efforts.
Mhm.
Also, the ones that they currently have is to help to see how we can either kind of convert it to a higher throughput um, um, method. So that's my role specifically. That's what I'm doing here. That's my primary role here at Lilly. So it's been very exciting. It's been a lot of learning. Yeah.
Yeah, that's great. So as you have recently transitioned from, let's say, an academic environment to industry setup, so what stands out in your opinion in these two different work environment? What are the differences?
Um. Good question. Um. My answers, these are based on the information I have for 6 months, right? I only started 6 months ago, so
Yeah, yeah, yeah.
obviously this is something that.
If you work here for a long time, you'll forget how academia was. So this is even better
So
that you still remember how your past life was, and you remember how different it is now.
yeah. Yeah, so I'll say that there are differences. Obviously, the science is the science. I think that's the one thing. The science is the same, you know.
Mm,
This is specifically at least for for scientists. I'll say the difference is at least for me is the access to resources is the first one.
okay.
There's a significant difference in the access to resources, and that's not a problem, especially
So do
for
you
big
mean you
pharma.
have more access now?
Yes, there's access to resources and just like basically anything you want. So like sometimes in my PhD, sometimes I want to do something maybe that we don't have the instrument or we don't have the money to buy certain reagents, or we have to wait, just a lot of things where we have to really just wait because there's no
Yeah,
money. Or
maybe
either there's no
needed
money or we have to look.
for,
Yeah.
yeah, maybe for one experiment you needed one other collaborator, and you had to wait for that.
Way. But thankfully, in a big pharma like Lilly, for example, I have structural biology experience, but like there could be a project that needs structural biology, but they literally have a structural biology team, and all you have to do is bring them in, and they do the work for you. They have an expression team. They also automation is also a big thing. So
Mm,
a lot of their things are automated, even the expression and the purification. So for lots of assays, plate-based assays, 96 well plates, they're doing these things in 96 well plates. And so you're talking ramping up this because you need to study as many as you can, right? Because this is the basic science, this is the discovery part, and so I think the resources is the good thing. Like I said, there's sometimes that I have to wait for my PI. Obviously, I understand the situation, but sometimes that might just be the bottleneck to finishing my assay or starting an experiment.
mm,
Um, in this case, again, that's not an issue. All I have to do is go in and order what I need. Obviously, as long as it's not, I don't know, 100K, but even if it's 100K dollars, as long as it justifies like what you want to do, that's something that is not an issue. So that's one thing. The other thing I noticed is I guess the team spirit. I'm not saying there is no team spirit in academia, but I would say that if you're in academia, it is completely okay if you're a PI or maybe a postdoc or a PhD student. It's completely okay to do your work. It's
mm.
up to you if you feel like you want to collaborate or if you feel like what I want to do, I need to talk to someone. You can decide to figure it out all on your own. I
Yeah.
think in a big pharma like Lilly, you can't really work in a silo. You have to work with a team. So if you're not a team driven person, you might have an issue with that, because you do have to have, oh my god, the meetings, you have to have a lot of meetings to discuss a project, so this involves, one project will involve hundreds of people. So
Mm-hmm.
that's the other difference, and I think that's also what makes the science faster, right? There's a business need for a project, so you have to make the science is good, but it's faster. The last thing I'll say is the um the way you do science. So in academia, you can have this wonderful idea, and yeah, as long as you can convince the NIH, they'll give you the money, right? So that's fine. In this industry, obviously, you have to align with the goals and vision of the company. It also has to be within the business need. So that's also something that people, or like, I had to adjust to. So you can't just wake up and just do anything you want. Like, you can't just have a wonderful idea, a random ass idea, and just do it. It has to align with the business needs. So that's also something. I guess that's why a lot of people feel like, oh, like, I don't want to work in industry. I want to have the freedom. You do have freedom, just like the freedom is kind of constrained to the company's business need. If they want to work on something that is, I don't know, neurodegenerative diseases, you have to work on that. Cancer or something, you have to work on that, right? So I'll say those are the three things. Three
Okay.
key.
Yeah. Really important points that you have talked about, team. And everybody should keep in mind when they want to move to
Exactly.
industry. Yeah, that's important point. So now, my next question is for someone who is a graduate student, or who is planning to graduate soon or have graduated recently. So if they have plans to go for biotech industries or pharmaceuticals like yours, so how they can make their profile a strong one? What should they keep in mind when they kind of arrange their CV, and what should they do?
Yeah, thank you. That's a good question. I feel like we can have an entire whole podcast for this, but I'll try my best and keep it to like 5 minutes. Um,
We'll
but
do that. But for now,
okay,
we can just briefly mention important points. Yeah.
okay, because like it's a whole thing, but we can have a whole masterclass on it. But I think that what people need to do, for people that are, maybe about to finish or just finished, I think the work that has to be done has to actually have started way before you finish. So what I notice is that a lot of people tend to just wait. And I get it, this thing is not taught. Nobody tells you as a PhD student or something, oh, this is what you should be doing. Nobody really teaches you that.
Hmm.
So a lot of people wait till maybe they are done, they
Right.
defend, and then they say they are looking for a job. So a lot of people don't realize that you can actually start working towards it. For me personally, I knew that I wanted to work in big pharma.
Hmm.
Maybe it's because of my background in pharmacy. I'm a trained pharmacist. So I
Hmm.
always knew I wanted to go. So it's okay that maybe some people don't know what they want to do. That's fine. But for me, I knew what I wanted, so I started working towards that. One of the things I know for working in biotech, pharmaceutical industry, or something like that, you have to work with people. I kept stressing team. You have to work with people. So you can imagine where your personality or the kind of communication, people don't stress that. I don't think it is necessary in academia. Like I said, I think it's important, but I think you can be fine in academia. In industry, you have to work with people. You have to work with different cross-functional teams. It
Hmm.
requires you understanding the people dynamics. That's actually how you can, if you go up the ladder, being a director, you have to manage people.
Yeah.
So the management part, it's very important. So my point is, I think that networking is very important. That's
Hmm.
number one. And that's
Okay.
something that you can start from any time. You don't have to wait. So a lot of people wait and then start networking. It's not easy to network. So for me, I started identifying people that I thought would be instrumental for my career
Hmm.
right from even when I was doing my master's. So
Yeah.
for a PhD student, I would say start as early as you can. When you go to a conference, PhD students go to conferences. A lot of
Yeah.
times they there, sit there and listen to seminars or listen to talks. That's fine. But I think that is also important that for me, if you go to a conference and you don't come back and you didn't make at least 3 to 5 contacts, I don't know, So for
You
me,
wasted your
that's not
time.
exactly. So for me, I'm like, you should be able to meet someone, you talk to someone. For me, I always try to identify people in the industry that maybe that do what I aspire to do. So I was
Mm,
always going to conferences. I read people's profiles. Sometimes even before the conference starts, I look forward to meeting people. I look up talks. Sometimes after the talk, I have to gather the courage to talk to people. So I usually establish that, and then I basically get that network because sometimes those are the people that may be instrumental to your
mm,
career. Sometimes when you're looking for a job, because that's the issue. When you're done, you can now go to LinkedIn and reach out to these people and say, "Hey, do you remember me? I'm actually looking for a job." And those are the people that might put in a good word for you. So
yeah.
that's one thing. The other thing is the approach to job applications. I don't have all the answers, but I think that the way, obviously, I also know that it's a very difficult time now. The job
Mm,
market is really, really hard. So even the best people are still having problems or issues with finding a job. I wanted to finish my PhD last year, December. But as of last year, December, I didn't have a job offer.
mm,
And I started looking for jobs last year, June. So it took me, as of December, I didn't have an offer. So I moved my PhD completion to May. So I actually got this offer from Lilly in March. So there's a lot. That's why I said you can have a whole thing about that. But like, there's a lot to also consider when you're applying to jobs. Should you apply to every job? I don't think so. I think you have to first of all do the homework and figure out what you want. For
mm,
me, it's easier for you to do the homework first. Figure out what you want to do or the kind of things you want to do, so that when you are looking at this job board, you can easily be able to weed out the ones that you're interested in and the ones that you're interested in. The other thing too is that the timing of when a job is posted and when you apply is very, very important. For an industry job, I would say that all the places I got 3 offers, all the places that I got these offers, I applied to these jobs within the first week when the job was posted.
okay.
Now
So,
I don't know if that's what led to the offer, but what I can say is I'm very sure that absolutely puts my resume up there because imagine a
mm,
job was posted a month ago and you are
wow.
applying to that job a month from now.
Okay.
Now I am in here, most HR people, when they post the job, most of the time, 2 weeks into when the job is posted, they've already screened for the for the candidates that they
Mm,
want to.
okay.
So by week
So
two,
that's a really important point you mentioned. I just want to jump in. So when if they are so ready to start interviewing or judging the application, so did you mention that you have graduated already or you can join anytime? So what should we do when in
yeah,
that
I see what you mean. Yeah, it depends on the role.
hmm,
A lot of academic roles, a postdoc. So I got 3 offers. I got an academic postdoc offer at St. Jude's Children's Hospital. I
okay,
got a postdoc, an industry postdoc offer,
okay,
and then I got this full-time offer at Lilly.
mhm,
So academic postdocs or academic jobs, they usually tend to be more flexible in
hmm,
terms of how long they'll wait for you.
okay,
My point is, yes, you can start looking. I didn't think that people should start looking when they are 6 months out. Ideally, I think you should start. When I say start looking, like really, like actively start applying 6 months before your intended day
hmm,
of completion. Right. So I started looking last year, June. I wanted to finish in December. By December, I didn't have an offer, so I moved it to May. I got this offer in March. So most companies, in my experience, a lot of companies will wait for you. But I
okay,
would say like the wait though, I'll say maybe, I don't know, maybe 3 months tops, 3 to 4 months. So
hmm,
I got the offer. So I applied for the role in January. I got the offer in March and
Mm.
obviously I was still not done with my PhD. So they were very flexible. So yes, you should tell them when they ask you. You should
Right.
tell them when your post defense or like when you are going to be finishing.
Mm-hmm.
So I told them I was going to be done in May, and I also told them that I wanted to do the ceremony. When I got the offer, I wanted to just do the ceremony and be done in Baltimore once and for all before I came down to San Diego. So I asked if I could start in June, and they were fine with that. So that's about, I'll say, maybe 2 to 3 months wait. So
Okay.
I'm very sure maybe if I had said I wanted to start in July, I'm pretty so it depends, is what I mean. Like they are
Yeah,
very
yeah,
open,
I understand. Okay, got your point.
depends on the hiring manager.
Right.
It depends on the hiring manager. It depends on how also depends on how urgent they want the position to start. But
Yeah.
like I said, most of the time they are willing to wait for you as long as it's not ridiculous, like I don't know, 6 months, 9 months.
Mm.
Um, I think they will wait. So I think you just have to be transparent. So obviously, if you're going to be done, I don't know, this is December. If you're going to be done in June, and you tell them you're going to be done in March, obviously, if they give you the offer and they expect you to start in March, then So I think you have to be transparent, and people that want you will wait for you.
I understand. Yeah, yeah, yeah. That's more practical. Yeah.
Yeah,
And
yeah.
so yeah, yeah. I interpret you. So do you want to add something additional about the application?
Yeah, yeah. So the application, I was talking about the timing
Mm-hmm.
of application. I think that's also something I noticed a lot of times. People, like I said, all the jobs that I applied to that were like at least three weeks to 1 month since it was posted,
Okay.
I
Yeah,
didn't
yeah, yeah.
I didn't get a call back. So I
That's
didn't.
really important point. Yeah, I didn't know that. I
Yeah,
think this information
very
will help a lot of people. Yeah.
exactly. Like I'm like I know this. Like I've gone through the process now and I've seen it. It worked for me. I'm like the week that in fact if you can apply within the first 2 days that job was posted. Obviously, it depends on when you see the job, right? So
Mm.
you kind of have to. It was a full time job for me. Every day I subscribe to Indeed, LinkedIn jobs. So every day in the morning I will go through the jobs. In the evening I go to the job. Before I sleep I go to
Mm.
just the company I'm interested in applying to. Every day I go to their career boards, I look at the job. So every day you have to be checking. So that's something on your own end. You have to do it. So that's how I was able to. Also, sometimes for this is also another important thing. When I identify like jobs or companies, I tend to try and see if I can find people that work in that like because sometimes people or hiring managers themselves will post the job themselves on LinkedIn. They'll be like, we're looking for, this. So
Yeah, yeah.
those people, I tend to follow them on LinkedIn. And then I just like, for lack
Mm.
of better word, monitor their posts, right? Because that's also how you will know when a post is out. And then
Right.
sometimes people repost. And also there are people in the industry that usually they have a very good network. So they always repost jobs from every,
Mm-hmm.
big firm. So it's just really following those people,
Mm.
identifying and following those people. That helped me a lot. And it
Right.
helped me kind of like, so my job, I was just scrolling through LinkedIn and I saw the job. And I'm like, oh my God. And then I looked at it, it was posted that day. And
Mm.
then I had applied for it the next day. Another thing that will be helpful
Have to
is
be
if
really prompt.
Mhm.
Mm-hmm.
Another thing that will be helpful because the thing is, imagine as an HR, you look through the pool of candidates, and let's say out of the 100 people that already applied in the first 2 weeks, you can already see 10 people that are very good. Because that's my point. Sometimes, though, sometimes for 2 or 3 weeks, maybe they haven't found what they're looking for. So in that case, that's a different thing. Where maybe if you apply after a month, they might
Mm.
still call you. But
Okay.
right now in this job market, is my point. In this job market, there are a lot of overqualified people. A lot of people are in the market looking. So for you and I, straight out of PhD, you're probably competing for an industry job with someone with years of postdoc experience. So that's the other thing I was going to say. How do you actually make yourself stand out? Like I
Mm.
said, this is the whole thing. You have to also make yourself stand out. You have to learn how to sell yourself as a PhD, right? Apart from what you do on the technical thing. What are you going to be bringing to the table? How do you sell what you do? That's something that I noticed that a lot of people are not able to communicate that. You can know everything about your PhD, but communicating that to maybe someone that doesn't know. How do I explain to someone that does not even know what dengue fever is? Dengue fever. How do I explain to them why it is important? So that's
Mm-hmm.
what they're always looking for. They're always looking for someone that can work with a team. So when you get into the interview, that's the whole thing. How do you actually pass through the entire interview? Most companies, there's a three-stage interview process where the first stage is the HR screening, and then the second stage is hiring manager call, and then the last stage, most of them tend to bring you on site. And for the
Okay.
on-site interview, you have to give a 1 hour talk.
Mm-hmm.
Then you have to meet with different people in the team, and so that's a whole day of interview.
Okay.
So it's also
Usually
a whole
interviews. Yeah.
yes. So that's the whole topic of like how do you kind of stand out because most times they
So
tend to bring three.
starting from the first call to the third interview, how long does that take usually?
Um, it depends on the company too. Depends
Mm-hmm.
on the company, depends on the team. Depends on the so I don't know for the three jobs, I would say for Eli Lilly, for example, I got the call. Eli Lilly was actually just 2 steps. I didn't get any HR call. It was
Okay.
the hiring manager that reached out directly. So they do their things a little bit differently sometimes where the HR screens and just passes the resume to the hiring manager, and then hiring manager is the one that reaches out to you directly.
Mm-hmm.
So I got the hiring manager email late January. The job was posted early January, like first week of January, and it
Okay.
was late January that I got the email.
Mm-hmm.
And then I had the interview with the hiring manager late January, and then I had my on-site interview um, late February. And
Okay.
then after my interview, I got the offer 2 weeks after my interview.
Okay.
So I got the offer in middle of March.
Mm-hmm.
So I'll say that whole thing took like what, like about 2.5 months. But from when you go for the on-site interview, it takes it because it depends on for me, they brought three, I think they brought three candidates on site. And
Mm-hmm.
I was the first candidate to come on site.
Okay.
So if you're the first person to come on site, it will take longer for them to reach out to you to get back to you. But if
Right.
you're the last candidate, it might be shorter.
Yeah, yeah, I understand. Because they
Yeah,
need to see everyone before they decide.
exactly. Yeah, yeah,
Okay. All right.
yeah.
Yeah, you mentioned really important points. Yeah, definitely. I think whoever will listen to this, they will be benefited. Yeah.
Exactly. And all this I just have to summarize. We can go into it, but like this is just a summary. I kind of just highlighted. I think like the most important things that I feel like people it looks trivial, like it's not anything. But that particular timing I said, like and when people try it, at least for me it worked. Like I noticed obviously you have to also be at least I'll say at least like a 50 to 60% fit for the role.
Hmm.
Um
Okay.
as long as they're fit for the role.
So it doesn't have to be 100% match.
No,
Okay.
no, it doesn't have to be. For
That's
this
important.
Eli job, I wasn't 100% match. I was looking for jobs like HDX, mass spec jobs. No, I think my fit here was about 70%. Some
Hmm.
of the aspects that I'm supposed to be doing here, I'm learning them.
Okay.
So it's not 100%. So I'll say people should not shy away from applying
Hmm.
to roles where you're not 100% fit.
I understand. Yeah.
And I think the network comes back. If you can use your network, that's important because
Hmm.
referrals might go a long way for you if you can go. So you know the concept of referrals uh, for jobs. So sometimes employees they can refer you internally. So that can
So
also kind of bring your resume.
yeah, the referrals do you have the include the referral in the application system or in the CV? Where should you include the
No,
referral?
so the referral system is basically so let's say you have there is a job that you're interested in Lilly And then obviously you know me, like we know each other. So you can reach out to me and you can tell me about the role. And all I need is the requisition number. Like and I know I just need the
Okay,
job
so
description.
okay,
So
that
I go
happens internally. Okay.
so I go into the system and I refer you, and then what you do is you get an email that somebody referred you, and you then use the link
Got it.
to apply. So basically, it will be seen internally that you were referred by somebody. So, like I said, it's not 100% guarantee, but
Mm.
sometimes that can help kind of put your resume or yeah, sets your resume,
Yeah, yeah.
right. Yeah. So it helps. So that's where the
Mm.
network I was talking about is, because what happens is that people just reach out to people randomly on LinkedIn, and a lot of people will not refer people that they don't know.
Yeah,
So that's why I'm talking about the network.
yeah, yeah.
So if
It-
someone that I know, someone that I met at the conference 2 years ago, and I remember having a conversation with them, and they reach out to me, I'll be like, "Oh my God, yeah, I remember talking to you. Yeah, of course, I'm happy to send me your resume, and I'm happy to refer you." So that's what I'm talking about the network.
exactly. Yeah. Networking should start way ahead of time, not after the application.
Yes, exactly. So, but I notice a lot of people tend to like they do it like kind of late in the game. It's fine. It's just like that's what I mean. I feel like these are low hanging fruits that at least for me, I think that worked in my favor.
Mhm. All right. Thank you so much, Juliet.
Of course,
These are really helpful suggestions. Okay, so we'll finish, but do you have any last minute message for our audience? It can be about research, career, anything.
yeah, yeah, I do have. I would say for research, I would say like to people that are out there, any PhD student, any PhD candidate. I think that they need to realize that the work that they do is important, and they also another thing I need to tell people, anyone listening to this, is that I think you should not tie your worth. Don't tie your PhD rather to your self worth. I think a lot of people are doing that, and unfortunately, in this job market, it is very easy to get depressed when you finish your PhD, and I get it. You finish your PhD and you're wondering why I'm not able to land a job. So and my point is, if you tie your PhD to your self worth, it's very easy to kind of get beaten down
Mm.
in the market. So my point is that's that. The work you do is important. Start early. It's never too late to start networking. It's never too late to start building your career plan in your PhD program. For people that are finishing, that are thinking about their career prospects, I think they should have a very open mind. I think this job situation and job market, it's revealing a lot of things to me. It's revealing that I don't think that academia or the academic environment prepares us for alternative pathways as PhDs.
Mm.
PhDs do so many things. PhDs can work in consulting firms. PhDs can work in investment firms. So it's your PhD. It's not necessarily what you did in the PhD. It's that process that you went through. That critical thinking, having to start a problem or ask questions and solve it. I think that's what everybody needs. So my point is, I think PhDs should start thinking big and thinking outside the box. There are a lot of opportunities out there that are not the standard academia or even industry. There are so many more opportunities out there. So it's just for you to talk to people and be
Mm.
open-minded. And I'll say people should just, don't give up the job market. Keep trying and you will get something.
Yeah.
Yeah, I think that's it.
Really good suggestions. Yeah, we have to be open-minded, and there are so many options, including the typical ones. We have many different career path that we don't even know about, but it's
Exactly. And obviously, you and I like read the one thing I appreciate CBI for, right? These are things that we discuss in CBI. We have those pizza talks where we talk about like alternative careers, right? I know we always have that, but that's something that people don't think about. So I think that's a good thing for us or for like people in the CBI program, because it kind of exposes you to things that you should be thinking about. Science policy, like a lot of things that
yeah.
PhD doesn't think about, but like they need PhDs in those areas. Yeah.
All right. Okay, thank you so much, Juliet, for coming to this platform. And I think you shared your research and a lot of important information about career. So thank you so much,
Of course.
and thanks to the audience for watching and listening to us.